What if I told you that a potential defense against one of the world's most feared bioweapons could be hiding in something you probably eat every single day? Well, today we're digging into some really incredible research that points to exactly that. It's a surprising defense from a very, very common source.
To really get why this research is so important, we have to rewind the clock back to 2001. Those anthrax attacks, where a simple white powder sent through the mail caused so much panic, sent a massive shockwave across the country. It really highlighted a terrifying vulnerability. And that, of course, kicked off an urgent search for new ways to protect people, something beyond the tools we already had.
Now, our usual strategy for fighting bugs is super targeted, right? You get an antibiotic that's designed to kill one specific bacteria. But what if we flip that strategy on its head? Instead of building a specific weapon for one specific enemy, what if we could just strengthen our body's entire security force, make our first-line defense tougher and more alert, ready to fight any infection that comes its way?
And that leads us to this big question. Can we basically train our own immune system, our body's general-purpose security detail, to just be on a higher state of readiness all the time? This whole idea is called non-specific immune stimulation, and that's really the key concept we're going to be exploring.
So allow me to introduce the potential hero of this story, beta-1,3-glucan. I know, it sounds pretty scientific and complex, but it's really just a natural type of carbohydrate fiber. You find it in the cell walls of things you see all the time, like mushrooms, oats, and yeast. And for decades, scientists have known it has this cool ability to interact with and kind of tweak our immune systems.
And while you can find it in a ton of common foods, the really fascinating part is that the specific beta-glucan they used in the studies we're about to dive into comes from a source that could not be more ordinary: baker's yeast. Yep, that's right, the exact same kind of yeast used to make bread and beer.
Okay, so how in the world do we know this stuff might work against something as serious as anthrax? Let's get into the evidence. And I really need to be crystal clear about this. All the data we're about to look at comes from preclinical studies. They were conducted in mice, not in humans.
You know, the way they designed the experiment was actually pretty simple and straightforward. The scientists took one group of mice and gave them an oral dose of this yeast-derived beta-glucan. Another group, the control group, just got a placebo. Then they exposed both groups to what should have been a lethal dose of anthrax spores, and they watched to see what happened.
And the results were, well, they were just stunning. In one study from 2002, the mice got the beta-glucan before they were exposed to anthrax. Now, in the control group, the ones that got the placebo, only half of them survived. But in the group that was pretreated with the oral beta-glucan, 100% survived. Every single mouse lived.
Okay, so that's incredible as a preventative measure. But what about as a treatment? What if you're already infected? Well, a study in 2003 looked at exactly that. They gave mice the beta-glucan after they were exposed. And the results were just as mind-blowing. The control group here had only a 30% survival rate. But with just a low dose of beta-glucan, that number jumped to 80%. And with a high dose, a staggering 90% of them survived.
I mean, this data is just unbelievable. 100% survival in one test, 90% in another. It really makes you ask the question, how? How did it work? What was this simple extract from yeast actually doing inside the bodies of these mice to create such a powerful effect?
So the researchers found the first big clue when they looked at these immune messengers called cytokines. The mice that got the beta-glucan had this huge spike in these signals. You can think of cytokines as the body's emergency alert system. They're basically broadcasting the message, "We have an invader," and that immediately calls immune cells like T cells and macrophages into action to fight the infection.
And what happened when all those alarms went off? Well, a later study actually measured the amount of anthrax bacteria left in the mice's lungs. The results were wild. The control group had over 5 million bacterial units. The beta-glucan group, they had less than a tenth of that. And get this, about 86% of the treated mice were completely bacteria-free, compared to just 41% of the controls. Their immune systems were just way, way better at clearing out the threat.
So we have this incredibly powerful effect, a massive improvement in survival, and we have a clear reason for it, an immune system that's been put on high alert. But what does all of this actually mean for us in the real world? It's so important to put these findings in context because this is an unbelievably promising, but also very early step.
All right, let's just wrap it all up. Here's what we know. We're talking about a simple supplement taken by mouth that comes from baker's yeast. In mouse models, it worked as both a preventative shield and a treatment for anthrax. The survival rate shot through the roof. And it seems to work by giving the whole immune system a general purpose boost.
But, and this is the biggest takeaway of all, these are animal studies. They show incredible potential in mice, but they are not human trials.
You know, that's exactly how the scientists themselves talked about it. This quote from that 2002 paper says it perfectly. This provides preclinical evidence. It's a powerful proof of concept, a really critical first step on a much longer road, and it absolutely means we need to investigate this more.
And that brings us right back to our huge question at the beginning. The evidence we see in these animal studies it's incredibly compelling. The source is as common as a loaf of bread. The potential is enormous. So could a simple component of baker's yeast really be a part of our future defense against a bioweapon? Based on this early research, that's a question we should absolutely keep exploring. Thanks for digging into this with me.